Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A level Biology Unit 5 Scientific Article for June 2023. These are the potential topics as well as questions that could be asked out of this article for question 8 of the June 2023 Unit 5 Biology paper. Let's begin with the article. The article is entitled Microbiota Gut-Brain Axis and the Central Nervous System. They begin with gut microorganisms. In paragraph 1, the human gut contains various microorganisms such as bacteria, fungi, parasites, and viruses, and more than 100 million bacteria reside in human gastrointestinal tract, which is 10 to 100 times the number of eukaryotic cells in our body. After years of common development with the human body, the gut bacteria have reached a mutually beneficial symbiotic state with the human body. In paragraph 2, gut microorganisms play an important role in promoting adult development and homeostasis. For example, they can affect human metabolic functions by decomposing the complex polysaccharides in food. In addition, gut microorganisms can regulate gut movement, the gut barrier system, and fat distribution. Gut microorganisms can affect immune function through the development of gut-associated lymphoid tissue and by preventing the colonization of pathogens, and they can affect the energy metabolism and mitochondrial function of the host. The intricate relationship governing host and microorganisms' interactions suggests that when this relationship is abnormal, the microorganisms may cause the pathogenesis of disease or promote the progression of disease. Therefore, recent research has focused on determining the diversity of these microorganisms to clarify the physiological roles they play and eventually to prevent and treat disease by controlling the microorganism species. Paragraph 3. There are three main methods for detecting gut microorganisms: the bacterial culture technique, the traditional molecular biology technique, that is independent of culture, and high-throughput sequencing technology. The former is mainly used for stool culture. This method is time-consuming, and the bacterial species obtained are limited. The latter two mainly isolate the bacterial DNA from the stool for the detection. The detection is fast, and the bacterial species are complete. The next part is about microbiota gut-brain axis, meaning microorganisms and interference in the central nervous system. In paragraph 4, the central nervous system is closely related to the gastrointestinal tract, and the CNS plays an important role in regulating gut function and homeostasis. In turn, the gut flora may affect the central nervous system and nerve cells, participate in the regulation of nervous system function, affect the pathogenesis and progression of nervous system-related diseases. Due to complex relationships between the gut microorganism population and the host, the authors proposed a new concept, the microbiota gut-brain axis. The microbiota gut-brain axis is the focus of recent research on the gut microecology. In addition to studies of the relationship between the gut microecology and neurological function, recent studies have emphasized how this relationship affects human health. In paragraph 5, the brain and gut can be connected through a variety of pathways, including the enteric nervous system, ENS, the vagus nerve, the immune system, or the metabolic processes of gut microorganisms. In paragraph 6, the vagus nerve of the body can control the function of multiple organs, such as the heart, the gut motility. The vagus nerve can also transmit peripheral immune signals to the central nervous system. The vagus signal from the gut can trigger an anti-inflammatory response against the sepsis induced by microorganisms. Gut microorganisms can affect brain function through the vagus nerve. After a vagotomy, the microorganisms will not be able to regulate behaviors. Continuing on. In paragraph 7, because gut microorganisms can directly affect the immune system, immune activation may be the pathway for transmitting microbial actions to the central nervous system. Microorganisms can also enhance the anti-tumor immune effect of drugs by promoting T-cell accumulation and transformation, and microorganisms are very important for the immune function of organisms. The immune system plays an important role in maintaining health by maintaining gut homeostasis. In paragraph 8, microorganisms can also cause neurophysiological changes in the host by producing chemical substances that bind to the receptors inside and outside of the gut. Paragraph 9. In addition, studies have shown that the microbiota may affect the central nervous system by altering adult hippocampal neurogenesis. The adult hippocampal and lateral ventricle have the function of generating new neurons. HN has a role in learning and memory and can have effect on the pathogenesis of many neurological disorder-related diseases and symptoms, such as epilepsy, depression, Alzheimer's disease, and Parkinson's disease. Next, they talk about microorganisms and brain function. In paragraph 10, the impact of microorganisms on behavior and cognition has been increasingly recognized. Microbial signals can regulate important functions of healthy human bodies, and growing evidence has demonstrated that many diseases are due to disturbances of gut microorganisms. Early studies in animals show that the introduction of single unique flora could lead to the development of anxiety-like behavior, and this change was accompanied by the activation of neurons in the brain that relied on the gut information transmitted to the brain via the vagus nerve. In paragraph 11, the change in gut microorganism found in sterile animals or with the use of probiotics, antibiotics, and colonization with fecal microorganisms can influence the cognitive function of the host. For example, supplementation with probiotics for a week prior to infection can not only prevent the microorganism disturbance caused by the infection, but also prevent the changes in cognitive behavior caused by stress. Liang et al., meaning Liang and his colleagues, found that probiotics could significantly improve the cognitive dysfunction induced by chronic restrained stress. In human experiments, functional MRI tests found that the activities in the brain regions that control brain memory and processing of sensation were altered after female volunteers consumed fermented milk that contained probiotics. The above various studies found that the stability of the equilibrium state of normal microorganisms in the gut was closely related to brain development and function. Microbiota, gut-brain axis, and neurophysiological disorders. In paragraph 12, schizophrenia is a neuropsychological disorder, and whole genome analysis suggests that immune-related genes may be changed in schizophrenia patients. Microorganisms and intestinal mucosal cells can regulate the changes in chemical factors, such as the pro-inflammatory interleukin 8 and interleukin 1, or the anti-inflammatory interleukin 10, and transforming growth factor B. The serum levels of pro-inflammatory cytokines in schizophrenia patients are higher than those of normal controls, and the level of serum inflammatory markers are positively correlated with the clinical symptoms of schizophrenia patients. In paragraph 13, gut bacteria can also produce harmful substances that damage the intestinal epithelial barrier, causing neurotoxic bacterial products and proteins to enter the circulatory system. Severance et al. Found that the concentration of antibodies against Saccharomyces cerevisiae was higher in the body of schizophrenia patients, and this genus was a marker of gut inflammation. An increase in the level of circulating pathogen antigen can cause the host to respond to its own tissue and cells through a form of molecular induction, and this response is the central process of autoimmune diseases. Compared with a normal population, schizophrenia patients have a higher probability of developing autoimmune disorders, and specific brain regions of schizophrenia patients, such as the hippocampus, amygdala, the frontal cortex, have higher levels of autoimmune antibodies. In the next part, microbiota, gut-brain axis, and neurodegenerative diseases. In paragraph 14, multiple sclerosis is a chronic inflammatory demyelinating disease of the nervous system, and its etiology is still unclear. Multiple sclerosis is associated with a significant increase in the number of cells that have immune response to the patient's own nervous system because gut microorganisms play an important role in the development of the autoimmune system and are associated with a variety of autoimmune and metabolic diseases. Therefore, it is speculated that gut symbiotic microorganisms play an important role in the susceptibility of multiple sclerosis. In paragraph 15, pro-inflammatory factors associated with chronic bowel disease can induce intracranial inflammation, lead to the death of dopaminergic nerves, and eventually cause the development of Parkinson's disease. The inflammatory changes observed in Parkinson's disease patients and PD animal models are associated with increased gut permeability. LPS is a gut-derived pro-inflammatory bacterial endotoxin that can cause changes in the substantia nigra, and it can act as a Parkinson's disease promoting substance. Similarly, Kashavarzian A. et al. Used high-throughput sequencing technology to examine the stool samples from 38 PD patients and 34 healthy individuals, and they found that the LPS system gene was significantly higher in PD patients than in normal controls. In paragraph 16, Alzheimer's disease is a degenerative disease of the central nervous system. Its onset is recessive, and its disease course is chronically progressive. The pathological markers of AD include extracellular beta amyloid, senile plaques, and intracellular neurofibrillary tangles. The number of maturation of microglia cells in sterile mice are abnormal, resulting in damage to the immune system and ultimately leading to the development of neurological diseases such as Alzheimer's disease. Cognitive behavior impairment is a characteristic of AD patients, and the influence of gut microorganisms on cognitive behavior capability has demonstrated the role of gut microorganisms in the pathogenesis of AD. In paragraph 17, the integrity of the blood-brain barrier is important for brain function and development. The inflammation caused by the changes in gut microorganisms will lead to changes in the blood-brain barrier integrity, which in turn affects brain function. Under normal conditions, LPS cannot enter the bloodstream due to the tight junction between intestinal epithelial cells. However, when the tight junction of the cells is disrupted and the permeability increases, LPS can enter the bloodstream and induce inflammatory response. Studies found that the plasma LPS concentration in AD patients is 3 times that of normal patients. Furthermore, intraperitoneal injection of LPS into mice can cause an A-beta protein increase in hippocampus, cognitive defects, and memory impairments. The increase of the inflow and the decrease in the outflow of the A-beta protein in AD patients caused the aggregation of the A-beta protein in AD patients, and this finding suggests a decrease in the capacity to clear the A-beta protein and an increase in the blood-brain barrier permeability in Alzheimer's disease patients. The increased concentration of plasma LPS in AD patients implies an impairment of the gut barrier function and increased gut inflammation and permeability, which further suggests that gut microbiota may participate in the pathophysiological process of AD. In paragraph 18, gut microorganisms can also affect brain functions through the synthesis of various substances. Serotonin is very important for cognitive function. 95% of serotonin is synthesized in the gut, and gut microorganisms play an important role in serotonin synthesis. The gut serotonin levels in sterile mice were 60% lower than the normal value. The use of serotonin reuptake inhibitor can reduce the beta protein levels in the brain, indicating that serotonin can reduce the formation of A-beta plaques, thereby reducing the risk of AD. In sterile mice, BDNF was significantly reduced, and this change was accompanied by cognitive function changes. Similarly, in AD patients, the BDNF levels in the brain and in serum were significantly reduced. The A-beta production and clearance in the central nervous system is a dynamic change, and some bacteria and fungi can secrete amyloid, resulting in an increase of amyloid levels in the central nervous system that disrupt the dynamic balance of the A-beta protein, which leads to the A-beta protein aggregation in the brain and a high AD risk. Therefore, an imbalance in gut microbiota may promote the development of AD by affecting intestinal function and synthesis and secretion of substances. The summary, which is paragraph 19, the interactive relationship between the brain and the gut includes neurological, metabolism, hormones, immunity, and other aspects, and changes in any component may lead to a functional change in the two interactive systems. The normal ecological balance of gut microorganisms plays an important role in the maintenance of this relationship. Microorganisms affect the development and function of the central nervous system through the microbiota gut-brain axis. The mechanisms of many CNS diseases are still unclear, and the discovery of this complex relationship, the microbiota gut-brain axis, has provided a new research direction for the study of CNS diseases that do not have a clear pathogenic mechanism. So this brings us to the end of the passage. I'll begin with the potential questions as well as the topics from which I expect some of the questions to come. Let's begin here. So from paragraph 1, I expect they will talk about bacteria. There is a possibility of talking about the common structures and functions. Remember, they have the nucleoid, the cell wall made out of peptidoglycan. They have the 70S ribosome, as well as the cell surface membrane. You also need to remember that some bacteria have pili, flagellar, plasmids, and so on. They could also talk about viruses because there is a hint about viruses from that paragraph. Remember, viruses have a protein coat. They have virus attachment particles, or what we call antigens. They have genetic material, as well as more other things. And also from this paragraph, they could include a question about methods of spread, how some of these microorganisms enter our bodies. So you need to visit the textbook on page 102, talking about vectors, inhalation, ingestion, direct contact, inoculation, fomites, and so on. You also need to visit barrier to infection, again in the textbook, page 102 to 103. They could also ask about benefits of gut bacteria. You need to remember that they help us to prevent the pathogens from increasing in number by outcompeting them or by competing with them for space as well as nutrients in order to prevent the number of pathogenic bacteria from increasing in our body or in our gut. They could ask you about features of eukaryotic cells, so you need to know that as well. From paragraph 1, I do not expect many questions because the content was focused on the introduction. Now, when we go to paragraph 2, they can ask you about how microorganisms in the gut decompose complex polysaccharides in the food. You need to remember that they can secrete enzymes, and then these enzymes break down components extracellularly, and then after, those components are going to be absorbed into the microorganisms. They can ask about the importance of gut microorganisms in regulating the movement of the gut. They could also ask about microbiota and prevention of colonization of pathogens, of course, like what I've talked about in paragraph 1. They can also ask how do gut microbiota affect the energy metabolism and mitochondrial function of the host cell. I suspect they could link a question about respiration on this because they talked about the mitochondria as well as energy, so a question on respiration could be linked. Then they can ask how can microorganisms cause the pathogenesis of disease. So you need to understand about chemicals that are produced by these microorganisms. They could be endotoxins, exotoxins, or other chemicals that are produced by these microorganisms that could act as antigens in order to trigger the immune response. So I also see the major point is either about toxins by the bacteria, infection of the endothelial cells by viruses, and resulting into cell lysis, and this can lead to increased gut permeability or inflammation of the cells, and this could induce immune response or activate immune cells. So there is a lot of these antigens produced by the microorganisms and how they can trigger the immune system, as well as how these microorganisms can enter into the blood through increased gut permeability. Now, when we go to paragraph 3, from paragraph 3, they can ask about bacteria culture as well as analytical techniques. So practice detailing the culture on agar, on broth, and using aseptic techniques. You need to revisit using a hemocytometer, dilution plating, optical density techniques in order to count the number of microorganisms. You also need to revisit the growth curve of bacteria and learn to explain each part of the shape of the curve that is produced. They could also ask about methods of isolation of DNA, but this can be combined with PCR, gel electrophoresis, microarray, and bioinformatics. So you need to learn the details of DNA isolation and production of fragments using restriction endonucleases. From paragraph 4 and 5, they can ask about homeostasis. How does the central nervous system regulate homeostasis? Here they could include about nervous control of the digestive system. They could also ask about neurotransmitters, how they are released. You need to remember that they bind onto receptors on the surface of the cells, then stimulation can occur. From these paragraphs, they also talked about how these gut flora affect the central nervous system. For example, it could be that these microorganisms can penetrate through the blood-brain barrier and therefore cause inflammation within the brain. Or it could be that substances are produced by these bacteria that can enter into the blood, cross the blood-brain barrier, and then trigger inflammatory response within the brain. So you need to know about that. They can also ask unrelated questions about the nervous system, like nervous control of the heart, as well as control on ventilation. Continuing on. From paragraphs 6, 7, and 8, there is going to be nervous control of organs like the heart, the lungs, the skin, and the liver, and so on. They can ask about microorganisms and how their presence causes inflammation, and then how that inflammation can trigger the immune system. So on that note, you need to know the details of specific as well as non-specific immune response. There is also some statement about microorganisms and T-cell accumulation. So on that note, they could talk about protein synthesis and how some chemicals produced by these microorganisms could lead to speeding up mitosis and thereby increase the number of cells that are produced. They could also connect these to gene expression and so on. You also need to know about how the immune system maintains health by maintaining gut homeostasis. For example, it clears away the pathogenic microorganisms in order to maintain a balance of the normal gut flora. You need to remember that microorganisms can affect changes by producing substances or toxins or chemicals that bind to receptors of cells and act as transcription factors, causing gene expression. And whether they produce a second messenger or directly enter the cell, they could participate in controlling the expression of specific genes. From paragraph 9, they will ask questions about Parkinson's disease, so you need to know the details, death of dopaminergic nerves, leading to less production of dopamine, which is a neurotransmitter. You also need to know that L-Dopa is used in treatments of Parkinson's disease, and that L-Dopa can cross the blood-brain barrier, and the body can use it in the production of dopamine. So they could ask about depression and serotonin, as well as the selective serotonin reuptake inhibitors, so you need to revisit your textbook and learn about these details. Now, in your textbook, you do not study about Alzheimer's disease, but they could connect a question on this because the passage has explained a little detail about Alzheimer's disease. So you need to remember that in this condition, neurons die and there is going to be memory loss. Another part I saw is they're going to ask about brain imaging. So you need to know how to compare and contrast between the different imaging techniques like CT scans, MRI, fMRI, as well as positron emission tomography scans. So they can ask about that as well. In paragraph 10 and 11, they'll ask about action potentials, generation of an action potential, propagation of an action potential across an axon. They can ask about reflex actions, the sympathetic versus the parasympathetic systems. They can also ask about how antibiotics work. You need to remember some are bacteriostatic and others are bactericidal. So remember the details of how bacteriostatic antibiotics as well as bactericidal work. And again, in these two paragraphs, they can talk about brain imaging because there was a statement about functional MRIs. They could also ask about how some of the chemicals negatively affect the nervous system, and this could be, of course, similar to what we've done already by binding onto specific receptors to interfere with neurotransmitter binding sites, and how that can prevent impulse transmission through specific neurons. From paragraph 12 and 13, here there was a statement about genes changing. So please revisit mutations. There are substitution mutations, insertion mutations, deletion mutations. You need to know the details of how these three types of mutations affect DNA as well as the produced protein. Since cytokines and interleukins are proteins, I believe they could ask about protein synthesis here. So you need to have deeper knowledge of protein synthesis from formation of messenger RNA to when the messenger RNA goes through post-transcription modification to produce the mature messenger RNA, and how that leaves the nucleus and goes to the ribosomes in order for the protein to be assembled. So they would ask about pro-inflammatory cytokines and how these can lead to inflammation. So their presence could lead to inflammation. So you need to know about that as well, meaning maybe these act as specific antigens that can be detected by immune cells. So again, there are going to be questions about specific and non-specific immune response. You need to know the details of everything. They could ask questions about how genes are sequenced, like exons or introns. They can include questions on whole genome sequencing, like the processes of isolation of the genes, random fragmentation, using electrophoresis like gel electrophoresis. They can also ask about microarray and bioinformatics. And I think they could include about genetic modification as a solution to help some conditions that have been observed. In these two paragraphs, there were statements about chemicals that are produced by bacteria, and again, endotoxins and exotoxins. They could ask about how those affect the cells of the body. In paragraph 13, they use an example of higher antibodies against Saccharomyces in schizophrenia patients. Remember that antibodies are protein in nature, so again, this could be connected to protein synthesis. And if these antibodies can act as antigens triggering the immune system to cause the inflammation, and how this can lead to autoimmune diseases among schizophrenia patients. because the immune system will attack body cells as well as tissues. In paragraph 14 and 15, there are statements about demyelination. So I suspect they'll ask about myelinated versus unmyelinated neurons. They can ask you to compare and contrast their structure as well as speed of nerve transmission. You need to remember that with unmyelinated neurons, conduction can occur all across the axon, and this takes longer. For myelinated neurons, conduction only occurs at the nosal ranvier, so it's going to be faster. And again, in these two paragraphs, they'll talk about stimulation of the immune system, again, specific and non-specific immune response, and how this can be connected to death of cells in autoimmune diseases. They will also talk about Parkinson's disease. There can be questions about the implications of the death of these neurons. On that note, you need to remember that people with Parkinson's disease have muscle tremors as well as rigidity. So, due to loss of dopamine or due to low concentrations of dopamine. So, a question connecting to muscle contraction can be posed. They can also ask about Parkinson's disease and nerve impulse transmission across the synapse. So, you need to know how an impulse is carried across the synapse. They could ask about, of course, treatment for Parkinson's like using L-DOPA. They can also ask about inflammatory response and bacteria toxins. And again, you need to remember some of these toxins can act as antigens that can trigger the immune system. And then they will ask about gene sequencing, again, microarray, bioinformatics, gel electrophoresis, PCR, and so on. And then about gene expression. I think you can see some of the things are coming up with every paragraph. They are definitely going to ask about questions on gene sequencing. They're going to ask about inflammation. There is going to be specific immune response, non-specific immune response, myelination, demyelination, Parkinson's disease, dopamine, and so on. They're going to ask about a lot of questions. Please, you need to fully understand about neurotransmitters and how they work across the synapse. In paragraph 16 to 17, again, specific and non-specific immune response. For the second part, based on the statement, they say the number of maturation of microglia cells is abnormal. This means a lot of these cells are produced, so mitosis is happening rapidly. They can ask about mitosis and the rapid replication of cells. This can be connected to gene expression from AS biology, so revisit epigenetic control. They'll also talk about questions about crossing the blood-brain barrier, and again, this can be connected to pathogens, medicines, as well as illegal drugs, and how these can cross the blood-brain barrier and affect the central nervous system. They can ask about what causes increased permeability of the endothelial layer of the gut. You need to remember that during specific immune response, infected cells can be destroyed. And if these infected cells are endothelial cells, these can lead to increased permeability. And if foreign chemicals or cells are detected in the bloodstream, inflammatory response can be triggered. They're going to ask questions connected to protein synthesis in connection to the excess production of specific proteins, and the consequence of this can be asked. Remember, they talked about A-beta proteins. If they're increasing in number and there is a lot of protein aggregation, that could mean the level or the speed with which these proteins are cleared is not as fast as the speed with which they are made. So they can connect these to epigenetic control, what causes excessive protein synthesis, and so on. From paragraph 18 and 19, they're going to ask about depression, serotonin, as well as selective serotonin reuptake inhibitor and treatments for depression. So you need to remember how this medication works. They could also ask about how neurotransmitters are made. And again, this is through protein synthesis. You need to know the organelles involved and the production as well as the secretion of neurotransmitters. You need to know the organelles involved in the production, packaging, and secretion of neurotransmitters. Remember about ribosomes, the rough endoplasmic reticulum, the Golgi apparatus, the secretory vesicles, as well as the process of exocytosis. And then they can ask about impulse transmission across the synapse. Please remember the details of that. Lastly, questions about how hormones affect organs they stimulate. And again, to emphasize, remember, steroid hormones enter the cells, but peptide hormones do not enter, but they cause formation of a second messenger. So you need to revisit page 214 to 215 of your textbook to fully remind yourselves of these processes. And again, because hormones are chemical in nature, I suspect that some of these bacteria toxins or the molecules produced or the chemicals produced by bacteria can also act like these hormones do. They could bind to specific receptors on specific cells and cause the expression of specific genes, leading to formation of specific proteins If possible, these proteins could aggregate or they could be produced in large numbers, and that could affect the central nervous system in a specific way. So this brings us to the end of this scientific article. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.